with a lecture by an ELSC member. Eran is familiar to most people here, but for the sake of maybe those who need an introduction, I will say a few things. So Eran's CV is 10 pages, and I have part of it here. I will not go through all of it. Eran did all his studies at the Hebrew University. He's a BSc in Life Sciences, Biology, and then Master's and the PhD, all here at the Hebrew University. He then went for a postdoc at the NIH with Tom Stelly, and he is faculty at the Hebrew University since 2007, and since 2016 he's a full professor here. I just want to mention that Eran gave, in addition to this talk, he gave a wonderful talk for the ELSC faculty last year, and it was very interesting, and I'm sure that this talk will be also very interesting. And so thanks, Eran, and you can see the title.
Toda Yoram. So thank you very much. It's a pleasure to open this series. Today I decided to talk about, so I'll show you some of the stuff that we're doing, but today I decided to talk about this topic, as you can see here, our work on pluripotent cells and neurodegenerative diseases, or basically modeling diseases using pluripotent stem cells. But before I begin, I have two before I begins. This is the first one. As part of the board member of the JBC, Jerusalem Brain Community, I promised to tell you this, that there are student-initiated workshops. You should take advantage of it. We pay up to $35,000 for such workshops, so you can invite people from abroad. These are initiated by 3 different students from, well, we prefer different disciplines, and the idea is really to initiate student initiatives. In addition, students who receive this funding will also receive an additional 1,000 travel grant to attend an international conference. So really take advantage of this. It's a unique opportunity that is not given by many institutions. So I think it's also a great opportunity to initiate and organize a workshop. So this is my first before I begin. The second before I begin, I just want to give you a very short, brief overview of the stuff that we're doing. So my lab is sort of a stem cell chromatin lab. We're doing mouse work as well as human work. So in mouse, we're doing very basic research, which is a little bit outside the scope of ELSC. We're studying reprogramming and differentiation, mostly neuronal differentiation, and we're looking at epigenetic and chromatin-related processes. And we're also doing human work. So in human works, we're using human pluripotent stem cells, and this is what I'll talk about today, and we're modeling neurodegenerative diseases. But we have a third topic that we kind of stumbled upon 2-3 years ago, a topic which we now call paleo epigenetics. It's a topic that I'm working with the lab of Eran Carmel. We have a joint collaboration on this, and if I have time today, I will tell you a few things about this as well. So basically, those who heard me, as Yoram mentioned, end of last year, this is the only topic I covered, so I'll not talk about it today at all, and I'm only going to talk about disease modeling using pluripotent stem cells. Now, because a lot of you are not familiar with this topic, I'm going to take it kind of slowly and introduce the concepts and maybe get to actual data. So I apologize in advance for those who really are keen for data embossed lecture. So I'm really just going to show the approaches and really just touch upon the data in the end. Okay, so this is the motivation. The motivation is basically that it's very difficult to study human neurodegenerative diseases. because of several basic problems. The challenges basically are, first of all, it's a very complex problem, obviously, and by and large, the studies regarding neurodegenerative diseases has been focused on animal models. And these, in many cases, do not model well the neurodegenerative diseases, of course, for several reasons. First of all, lifespan, the size of the brain, and the late onset issue. There are various different issues with animals that do not recapitulate well neurodegenerative diseases. Of course, the brain is inaccessible, so this is why we use animal models. And even if in the rare cases that we get brain biopsies from patients, these are usually, of course, not healthy patients, right? There's a reason why this brain was open to begin with. And whereas we can obtain relatively easily blood, liver, almost every organ, the brain is the least accessible organ from human donors. And again, if you have a brain tumor, it will be removed and you can get a sample. But this sample is very precious, and even when you get it, neurons, as you know, do not divide. So there are no neuronal cell lines. Very difficult to work with human neural cell lines. The neurons are post-mitotic, so once you get to a neuron, that's pretty much it. So I'll try to show you and convince you why pluripotent stem cells can be a good model to study neurological diseases. So the diseases that we're focusing on in my lab are a series of diseases called poly-Q disorders, and which are caused by a CAG expansion in the DNA. So just briefly, we have several genes which contain CAG repeats. CAG is a triplet DNA codon that encodes for the amino acid glutamine. And in healthy individuals, in most people in this room, we will have a stretch of about 20 glutamines in several different proteins. It's not clear why we have these repeats, but it's probably advantageous at some sort. And it's also, we have about 9 poly-Q disorders. I'll show you in a minute. But the bottom line is that 20 is healthy, and if it's more than 20, 40, 60, and above, sometimes it can reach more than 100 Qs. This is the disease form. And we think we know also the mechanism of expansion, but it's really not the topic of today. So we have healthy individuals and we have those disease-bearing individuals. Now, another thing about poly-Q disorders is that if you have a single allele mutated, you are going to die from the disease. So they are dominant diseases. Okay, so if you have one allele mutated, you're going to develop the disease, and in 8 out of 9 cases, you're going to die from the disease. So the penetration of poly-Q diseases is 100%, and it's a dominant mutation. This is the reason also we decided to work on these diseases, because there is no ambiguity. Many diseases such as Alzheimer's, Parkinson's, which eventually we're also going to get into, you don't know, there's a chance that this particular neuron is sick, but it's not 100%. With these guys, you know that if it carries the mutation, it's a sick neuron. So it was very helpful for genetic studies. So these are the 9 diseases. I'm not going to go into their names. You can identify that many of these are spinocerebellar ataxias. And we have so far modeled 2 of these. Huntington's disease, this is a prototypical poly-Q disorder, the most known one, the most common one. and another rare disease called spinocerebellar ataxia type 3, or Machado-Joseph disease. So Huntington's disease is caused by polyQ expansion in the Huntington gene, and the MJD disease, Machado-Joseph, is caused by expansions in the ataxin 3 or MJD1 genes. So one of the thoughts were maybe the polyQ itself has some kind of structural moiety to the protein, but you can see that actually this is basically the open reading frame of the gene, basically the coding region of the proteins. Okay, so we're now looking at all the 9 different proteins projected on the same line, and we're just looking at where the repeat is, and you can see that basically each one and every of these 9 disorders, they have the mutation in a different part of the gene, so it's not common to all these diseases. So Huntington's disease has an incidence of about 5 to 100,000. It is mostly starting and affecting the striatum, and the most affected cell types are these GABAergic medium spiny neurons, striatal GABAergic medium spiny neurons. So it's a typical cell type. And curiously, these 9 diseases, they affect, so the mutations affect completely different genes, different pathways. However, they all kill neurons. The mutation, for some reason, are always manifested in the death of neurons in the central nervous system. And this is one of the puzzles that we don't understand why. And in Huntington, these striatal GABAergic medium spiny neurons are dying, but for example, in MJD, the other disease that I just mentioned, Machado-Joseph disease, it's the cerebellar granular neurons and Purkinje neurons that are dying. So there's different specificities to the susceptibility of these mutations. So as I said, the gene affected is Huntington. And here you can see just a post-mortem brain of Huntington's patients with basically a deteriorating striatum. So the goals in these projects were to establish pluripotent cell lines carrying Huntington's disease mutation. And the idea was, of course, to generate relevant neurons or neuronal progenitors or a neuronal system that is human specific. Okay, so first of all, to establish pluripotent cell lines carrying specific mutations of these diseases. And one of the big issues until recently in the field was the controls. So what controls are you using? You have, let's say, cells from a person or cells from a specific embryo, and then you have healthy cells. So can these actually be compared? And there was a lot of noise that was introduced inside the literature because of these sort of non-isogenic comparisons. So the idea was to actually produce isogenic cell lines. So we have the mutated cell. The idea was to correct the mutation or otherwise take a healthy cell and introduce the mutation. So we have the exact same genetic background, differing only by the mutation itself. And then establish a reproducible neuronal differentiation protocol, which proved to be extremely challenging, but I'll show you some of the data. And then characterize and compare the neurons in vitro and in vivo. So here we can really sort of try and analyze molecular pathways and gene expression programs that differ due to the mutations themselves. Okay, so regarding pluripotent cell lines, there are 2 options, either using induced pluripotent stem cells or embryonic stem cells. So I'll introduce the 2 concepts very quickly. Embryonic stem cells are derived from the developing embryo, not to be confused with somatic, mature, or any other stem cells for that matter. Okay? Embryonic are unique in a way that they can be derived from the blastocyst. It's about 1 week after fertilizations in humans, about day 4 in mice. And all the stem cells that we have in our body, be it blood stem cells or kidney stem cells or other mature somatic stem cells, are completely different types of stem cells which can only produce a single type of cell, usually, or different types of cells, but usually confined to an organ, blood, liver, etc. Embryonic stem cells, because they are derived from the early embryo, this is the stage where the cells basically have not decided what they're going to be. Okay, so it's the first stage where we can distinguish two types of cells: the outer cells. These cells we know are going to be the placenta in mammals, and the inner cell mass. The inner cell mass are going to be us, basically. So each and every one of these cells possesses the entire potential to become any different cell of the body. So they have this great potential, and if you can grow these cells successfully in culture, you have a line of so-called human embryonic stem cells. Now, in order to model a disease, so of course if we do this from healthy embryos, by the way, this whole field started, or the idea behind it started in the late 70s and 80s when in vitro fertilization started. Of course, we don't have access to actual embryos, only in vitro fertilized embryos. And Louise Brown was the first IVF, in vitro fertilized, successful IVF child. She was born in 1978. So early 80s, there are now IVF clinics around the world, and there are superfluous embryos which can be used for research. And people actually have tried to grow these for many years. And only in 1998, the first embryonic stem cell line was successfully cultured. So it's a relatively new field, okay, about less than 20 years. And in order to use a disease-bearing human embryonic stem cells, you have to derive these cells from a disease-bearing embryo. So how do you have an embryo with a disease? Of course, most of the embryos of IVF people are healthy. So there's a procedure called Preimplantation genetic diagnosis, or PGD. So a couple where one of the parents is bearing a dominant, let's say a dominant mutation, such as Huntington. He is basically a ticking bomb, because he knows, let's say he's now 25, 30, 35 years old. He's still healthy, but he's probably going to die around 50, 55 from the disease. It's a terrible disease, by the way. It takes about 10, 15 years to succumb to the disease. But the patients, they start with this chorea, which is uncontrollable movement and gait, and they cannot eat, and later on they cannot walk. Eventually they die. So of course they want healthy children. So in order to obtain healthy children, they elect to undergo a process of in vitro fertilization. Of course, they don't have, usually it's not a problem of fertility. They don't have fertility issues, but they undergo IVF in order to screen the embryos. So this is the procedure. It's a procedure called Preimplantation genetic diagnosis. What the IVF clinics can do is pluck out a single cell from the 8-cell stage embryo. Okay, so this is when after fertilization, the egg divides into 2 equal cells, and then there's a division, another division, 4 cells, and another division. It takes about 24 hours for each division. So this is about day 3 after fertilization. You have 8 cells. This is a very early embryo. At this stage, you can pluck out 1 cell from the embryo, return the embryo to the incubator to complete development until the blastocyst stage, transplant the blastocyst back into the womb of the mother, and the embryo will be healthy. So the deletion of 1 cell at this stage does not harm the embryo. The first kids that were born with this procedure are already 16 years old, more or less, and they are healthy so far. So their development is fine, and everything seems to be going well. Okay, so this is a procedure that does not affect health in these embryos. So what these couples are doing, they're undergoing PGD, basically screening for embryos. When you undergo IVF, the female gets hormone shots, and then instead of producing one egg, she usually produces more than 10, 15, and more, depending on the fertility of that particular individual. But you normally have, it's a normal procedure to have a dozen embryos or so for a common IVF treatment. And then you can pluck out a single cell from each and every one of these embryos and test it using a simple PCR or FISH, just to screen for this particular mutation, in our case, Huntington. Okay, so you know that the father, for example, has the mutation, and on average, half of the embryos will contain the mutation because it's a dominant, okay? And so you'll have healthy mutant, etc. The healthy embryos can be frozen for future reference or put back in the womb for treatment, transplanted into the mother, and the mutation-carrying embryos can be used for research. So we can get those embryos, and then we have a disease-bearing embryo. And these embryos, of course, are otherwise discarded. Okay, and then you can derive human embryonic stem cells from these embryos, and then you have a disease-carrying human embryonic stem cell line. So actually, we were looking for human embryonic stem cell line with Huntington's disease mutations in Israel. We found none. We started looking in all the IVF, and this is actually, it's strange because there are quite a lot of HD patients in Israel, but I don't know why no one of the couples underwent, or at least didn't sign consent that they will they approve to donate their embryos, but we didn't find any such embryos in Israel when we started this project. Now there is one line that we got from Sha'arei Tzedek, from Racheli Aigesh. This is a new line, just about 8 weeks old. But when we started this, we did obtain lines from the IVF clinic in Sydney, Australia. Now they are actually sold under a company called Genea, but at the time it was just a research collaboration, and these were the repeats. So here you can see the CAG repeats of the two alleles, 1240, 1746, 1748, 2345, etc. And two of these lines we were able to grow successfully. So they are, again, heterozygous. By the way, homozygous for these diseases is exceedingly rare. I think only 2 have been reported so far in the literature. So all of these patients are heterozygous. But again, it's dominant, so these patients die from the disease. So the other way, other than obtaining cells from the actual embryos, there's now another way of creating pluripotent cells by reprogramming. So we now know that you can take a mature cell, be it a skin cell or a blood cell or even cells from the tip of your hair, and you can reprogram them back to a pluripotent stem cell-like state. So actually the father of this idea, at least the living father of this idea, is John Gurdon, who thought about cloning experiments, he and others, but he was the surviving researcher to collect the Nobel Prize, and he was the first to clone a whole animal. And the idea was basically whether a mature cell can reproduce a whole animal. So just in short, the experiment of the original cloning experiment, by the way, it was 1958. He cloned the frog, Okay, he took a fertilized. He used frogs, by the way, because the oocytes are about an order of magnitude larger than human oocytes. They're about 1 millimeter. You can manipulate them with your hands without using fancy microscopy. So he took a fertilized egg and he emptied it, no genetic material. And he then took a nucleus from an intestine and transplanted it into the egg, and then put the egg back into the frog. And these tadpoles were born in 1958. These tadpoles contained cells that were, until a couple of days ago, intestine. Okay? From a single intestine, a single intestinal nucleus produced a whole organism, basically. And in 1962, they managed to get healthy frogs. It took them 4 years. And this was the same year, 1962, that Yamanaka was born. And they both shared the Nobel Prize recently, 5 years ago, in 2012, for these achievements, okay, for the idea of reprogramming, either by somatic cell nuclear transfer or cloning. And again, just as a side note, in 1962, frogs were cloned successfully. It took more than 30 years. In 1996, Dolly was cloned. Okay? So it took more than 30 years to actually achieve the same feat in mammals. Many researchers actually left the race because they decided that it's simply not possible. But a very stubborn Ian Wilmot, a Scotsman, he was really, really stubborn, and eventually created Dolly. Now, Sir Ian. Okay, so, and what Yamanaka did, again, he was born that same year as these frogs. He did a very naive experiment. He basically, so this is a cell paper with a single author, either than Yamanaka himself. And actually, it always saddens me to see that Takahashi gets absolutely no credit and Yamanaka gets everything. So as a student, if you have a single author paper, really, you should get more credit for it. So anyway, what they did is they basically asked whether you can reprogram cells directly. So they forced the expression of proteins that are usually present in pluripotent cells and are sort of downregulated during differentiation or are less abundant in differentiated cell types. And they use viruses, doesn't matter all the technical details. The idea is that by expressing proteins in somatic cells, they were able, in a matter of several weeks, to reprogram these cells back to a pluripotent state. This was 2006 in mouse and 2007 in humans. And they produced these cells that they nicknamed IPS cells, induced pluripotent stem cells. And there was a lot of skepticism in 2006 and 2007, but in 2009, mice, whole healthy mice, were produced from IPS cells. Okay, so these cells that were until recently a skin cell, reprogrammed back to an embryonic stem cell, transplanted back into an embryo, produced chimeric mice without getting into the molecular biology, and eventually produced mice that contain only cells which were once a skin cell. So these are kosher cells, kosher pluripotent stem cells, which means that it's probably working for human as well. So because we cannot test it, we cannot really say in human, we can never say if it's really pluripotent because we can never generate humans from these cells. You cannot do this experiment. It's unethical. Don't try this at home. So we then generated human IPS cells from Huntington's disease and Machado-Joseph disease, but I'm just going to concentrate today on one disease for the simplicity. So people actually have done it before us, Lisa Ellerby, and then us, and then Mahmoud Pouladi, although you already published this. So all of us basically obtained the cells from the same repository. There's at the National Institutes of Health, NIH, they have a very, very large collections of cells from patients. So basically, each patient that go through the NIH, they ask him or her if they are willing to give them some blood or a skin biopsy. Most of them do, and they basically have a collection of almost any disease that exists today. So, therefore, this is the reason we both have the exact same cell line, because these were obtained from 180Q patients. This is the longest stretch that was reported so far. It's the late of onset is 6 years old. And I actually didn't mention that the longer the repeat, the younger the patient will start symptoms. And this is a late onset neurodegenerative disease. It usually starts at the age of 40. But when you have such a long repeat, it starts very early. It's a juvenile form. Again, very, very rare, maybe 5 or 6 cases in the world. So basically, we now have these two models. We have iPS cells, and again, this is simple, right? You can take a skin biopsy from any patient, and then reprogram the cells. So within a matter of a couple of months, you can already obtain these cell lines, and these are very rare. Okay, so there are maybe 2 or 3 places in the world which would even have these. And actually, for the other disease, so Machado-Joseph disease is about an order of magnitude rarer than HD, and we have cells from Eran, from Benny Rubinoff, which is the only one that we know of that exists today in the world. So to our knowledge, this is the first and only human pluripotent cell lines carrying the MJD disease. Okay, so once, just again as a side note, once you obtain these cells, you basically have to sort of test that they're pristine, kosher cells, just to make sure that no genetic aberrations, that they self-renew like embryonic stem cells, that their expression profiles mimic that of embryonic stem cells, that they differentiate. You have to sort of do in vitro and in vivo differentiation to make sure that you can generate endoderm, ectoderm, mesoderm, and also in vivo transplant them in skid mice to show that they can produce teratomas. So after these tests, you can call them pluripotent. Okay, so the next step was to produce isogenic cell lines. So this was very, very difficult until 3 years ago. But in 2013, the CRISPR era began. And if you haven't heard of CRISPR, something is wrong with our teaching program. So CRISPR-Cas9 is a system that bacteria use to fight against viruses, and it's a very, very simple method to direct a double-strand break in your DNA. So Feng Zhang at the Broad Institute and George Church at MIT, they both published now, I don't know, cited 10,000 times papers in Science back to back, and they show that you can use this system. It's a Cas9, it's a single protein that recognizes a RNA-DNA hybrid, and it cuts in a specific location. It doesn't matter, again, the details, but the idea is now you can transfect your cells. You can introduce a short piece of RNA together with this Cas9 protein and basically cut your DNA anywhere that you want. So this is really a revolution in terms of the ability of molecular biology tools. So now we can use this technology to basically cut and correct this mutation. It's actually much more complicated than this figure. It took about 2 years because of sort of technical details. Again, I don't want to get into it because this is really hardcore molecular biology. So you basically screen for many clones to see if you were able to correct. You're using the wild type allele to try and replace the mutated allele. So for those who are more privy with molecular biology, you basically have 2 bands. That one is the mutated, the longer band, and a wild type band, and then you're looking for only a wild-type band after the correction. And then you can also test this at the protein level using Western blots for either the Huntington gene or the polyQ itself. And then you can, again, screen at the protein level for isogenic cell lines. So this is the idea, and this is what we're doing as controls. Okay, so if this was relatively difficult, this was notoriously difficult because actually here we were kind of relying on pre-existing protocols. There were two papers that were already published where they showed that they can produce GABAergic neurons or striatal GABAergic neurons, because these are the medium spiny neurons of the striatum, but it really didn't work very well in our hands. So these are the protocols. But so Dorit, then a postdoc in my lab, started really to fiddle around with all the possible conditions, and about 5.5 years later, she was able to achieve these DARP32 positive neurons, which are presumably these the neurons that we were after. So you start with this PAC6 positive, TAJ1 positive early neurons. This takes about 3 weeks, and then over a course of about 2 or 3 months, you can generate these DARP32 cells. So this protein is presumably expressed in the neurons that we're after. But from a practical point of view, we realized that many of the experiments that we're going to do are not going to be in these endpoint neurons, because it's really a very, very cumbersome project. So we kind of diverted our attention to earlier types of neurons in order to be able to achieve things in a PhD time scale, right? Otherwise, it would have taken...
This was done on embryonic stem cell or on
so both. We always do both, yes. So another sort of method, which was again quite a shock. This was published 6 years ago, I think, in Nature by Marius Wernig, was that you can actually use factors, just like Yamanaka used a few proteins to direct fibroblasts into pluripotent cells. You can actually direct fibroblasts directly into neurons. So instead of going through the pluripotent state and then back to a differentiated neuronal state, he did the screen with several different proteins, and eventually they came up with 3 factors which direct almost any cell type directly into a neuron. And a few years later, a GABAergic neuron came out, a striatal GABAergic, and actually Moria in my lab tried this protocol, and actually it works, and it's reproducible, and it's nice. Again, it takes, this is day 16 neuron, but it really expresses the right proteins, and it looks like a neuron. So we sort of have these 2 types of neurons that we can work with. And the advantage of using this type of neuron is that you're basically, you're not going through a pluripotent state, and there's always this, when you start with a pluripotent state, there are always health issues, safety issues concerning your cell lines. But again, we can talk about it later. So to characterize the cells, we actually teamed with Yossi here, Yossi Arom, and with Vitali Lerner in his lab. We wanted to patch these cells to see that they actually contain actual potentials. So this is just to show some of the spontaneous neuronal activity that some of these neurons generated, and also we were able to show evoked potential. And actually another experiment that Vitali did is actually to use PTX just to see if they can, if they have GABAergic channels, basically. And so again, it doesn't mean that they're actually GABAergic, but this was the best experiment we were able to do. But these cells do express the correct markers, and also they show fire potential. And at least there should be neighboring cells that are GABAergic. So basically, if you use PTX, you get rid of this action potential, and if you wash it, it goes back. So this is, of course, I'm showing you some of the best neurons that we have. We have a collection of neurons, but at least some of them show the characteristics that we're after. So we can, I think we can say with confidence that we can generate neurons that look like neurons, they express the right markers, and they have action potentials. So a more recent way to now work with differentiation methods and specifically neuronal differentiation methods is differentiating these cells into three-dimensional structures. So far I told you about our sort of very difficult attempts to generate two-dimensional structures or uniform layers of neurons. But there's a way to actually differentiate these cells into three-dimensional structures, and this is a paper that was published 3 years ago by Knoblich and Lancaster, and they basically show that you can, again, within weeks or months, you can obtain, using the correct conditions, etc., tissues that sort of mimic the layered structure of the brain. So this was very appealing to many of us who were doing disease modeling and trying to get to neuronal tissues that is somehow relevant to get a better, maybe, model of the disease. So we started fiddling around with these differentiation protocols. So you start by generating these early embryo-like bodies, and then after a couple of weeks, you generate these funny-looking structures. But when you start to you section these and you stain, and this is just about a month into the differentiation, you see nice structures inside that look really mimic like layered structures of the brain. And yeah. No, absolutely not. So actually, they are extremely heterogeneous, and I think this is key, because now, from a molecular point of view, you can actually disperse this into single cells and obtain the cells that you want. Because one of the problems with using monolayer or single or directed differentiation is that you don't really have the environment of the cell, and I think this is a major caveat in really modeling these diseases. So here, I think we have again, it's very early. This is the pro. Of course, we're not mimicking a fully developed, mature brain, but at least from a developmental point of view, you obtain all the early neuronal markers that are there in the developing brain. So actually, to expedite this, we're also testing. This is a project that we're doing with Kobi Nachmias here in the institute, is to use endothelial cells, and these actually they do two things. One is that they form these structures that mimic blood vessels, although there is no blood in the system. We do realize that, but also they form a scaffold which for some reason helps our cells to differentiate much faster. So we get this we get this self-organization that is more rapid than without the rat endothelial cells, and also because we're using rat, it's very easy later on to separate these two populations. And one of the problems, again, this is just, I'm showing you sort of experts from some of the stuff that we're now just starting to do. One of the problems with doing these cellular aggregates or three-dimensional differentiation is that again, it's very cumbersome. You have to do one by one. And if you want to collect a lot of material, it's essentially impossible to do. So we started using beads, polymer beads, on top of which our cells start to form these three-dimensional structures. So together with Eran, who has a microfluidic device, we can basically encapsulate these polymer beads inside these microfluidic droplets, and basically use large-scale self-organization of these organoids, and this is something that we're just starting to do, but I hope it will become a reality in a few months. Okay, so one of the problems is that, of course, neurodegenerative diseases are late onset. It sometimes takes 50 years for a disease to break out, whereas the embryonic stem cells that we're using are basically at the age of 0, right? We're rejuvenating the cells. We're starting with very, very early undeveloped material. And this is a major problem in the field, sort of the elephant in the room, and sort of people ignore it. But one of the ways to sort of try and expedite the process of development is, and this is a paper that was published by someone else, but the idea was to use premature aging disease, or progeria, in order to expedite aging in the cellular system. Okay, so amazingly, I think, this disease is caused by a single mutation in a single gene in a dominant fashion. Okay, a lamin A gene, maybe some of you heard of it. Lamin A is a protein that you can see here. Here you can see lamin B, but lamin A looks exactly the same. Okay, it's a nuclear envelope protein. This is the nucleus of a healthy nucleus. Now, in these progeria patients, this is an 8-year-old child who almost looks like 8-year-old, and the lifespan of these children is about 13, 14-year-old, and all their organs, everything, they have accelerated aging in basically every single type of their body, less so in the brain, but at the cellular level you see advanced aging. So and also, if you take cells from 80-year-olds or 90-year-olds, you see more or less the same shape of the nucleus. So it really, you can really use this to model. So what happens? Again, I told you this disease is dominant. So if you express lamin A, the dominant form of progerin, the mutated lamin A, you can actually expedite aging in a cellular model. Because this is the wild type lamin A, and this is the mutated form of lamin A, and it causes the cells to be sick in progeria-like syndrome. Okay, so these cells are now, they feel like they're 80-year-old. And one of the interesting things, so of course there are lots of caveats. This is just one approach that we're taking. We can discuss why this is bad, but there is one result which I want to share, which is it's really kind of interesting. If you compare wild type and HD neurons, okay, so you differentiate the cells, the wild type cells and the HD cells, and you compare the expression profile of these two neuronal types, there isn't much of a difference. There isn't much of a difference. By and large, you don't see a lot of differences. There are some, but it's quite disappointing. But if you now express progerin, if you age your two systems, we're not looking at the aging effect per se, which also has the issues, but we're now aging the two systems in a similar manner, you start seeing differences that are much, much bigger than what you saw earlier. So progerin expression enhances the differences in gene expression that we otherwise don't actually see. So again, from a modeling point of view, it's very powerful. And then you can start doing candidate analysis and look for pathways that are altered in your HD cells, and these are just examples. Maybe I'll focus on one particular example, but I'm running out of time. So another thing that we're now starting to do is actually use single cell biology, basically single cell RNA sequencing, to try and see the population heterogeneity that we have in our system. So single cell biology offers two things. First of all, you can separate different cell populations, so you can now see the different types of neurons that you have if you're starting from a brain organoid or from a two-dimensional structure, and you can compare, of course, your healthy cells with your HD cells, etc. But also, single cell gives you the heterogeneity. And this is what Matan Sorek in my lab is doing. So he's asking whether neurodegenerative and neurodevelopmental diseases, he has an hypothesis that instead of just showing differences in gene expression that are either up or down in a particular gene or pathway, maybe in general things are just going awry, right? So you'd see things up and down at the same time. And this is in fact what he sees. So he has this correlation matrix where he's looking for correlations between two genes running over all the expressed genes. So the more this is shifted to the right, the more heterogeneity you have in the system. And here you have two different corrected cell lines and two different mutated cell lines. So basically, you're shifting your system back to a more regular or less variable state when you correct the disease. And of course, this is something that we are really interested in doing, but it's very difficult just from a technical point of view. So we started with a large scale, with a small scale proof of concept experiment. Now you can test drugs. If you have some kind of cellular phenotype or even molecular phenotype, you can see what these different drugs are doing and whether you can use these cell lines to actually screen for drugs. And in one case, and this is just one pathway that we're looking at, this is polycomb EZH2, is a gene that mediates lysine, methylation, and histones. I'm not going to get into it, but you can see that these are the healthy samples, these are the mutated samples. You can see that the gene is much higher, and these are the healthy iPS cells and the mutated iPS cells. So in both systems you see it higher expression, and when you treat with the drug, you get this very nice partial rescue. So I think I won't have time to talk about paleoepigenetics, so I'll just thank my lab. So a lot of this was done by Dorit, who really initiated the differentiation experiments. I've shown you results by Morian. Now Wala is doing some of the HD work in my lab, and Matan is doing the single cell RNA sequencing experiments together with Joshua Levin at the Broad Institute. Actually, without their help, it would have been too expensive. And thanks to Yossi Aron for all the electrophysiology. Thank you, and we'll do the other projects sometime in the future.
About the PGP cells. Actually, the state of Israel pays for PGP to separate healthy from unhealthy embryos, and it's used routinely when the testing is simple, like muscle diseases that would only occur in males because it's chromosome X, and that is used routinely. So perhaps simplifying the analysis could help to get this into more use in Israel. It's a really important technique. About the single cell sequencing, is the depth of sequencing sufficient when you look at one cell?
Okay, so this is the question is regarding the depth of sequencing. I actually removed the data where the depth is not right. So basically there are two ways to perform single-cell RNA sequencing, and this is why I said that it would have been too expensive for us, because what we are actually using is SMARC-Seq2. Now, SMARC-Seq2 is not a microfluidic system. With microfluidics, if you want to do thousands of cells simultaneously, you get, I think, with good accuracy, a few hundreds of transcripts, maybe up to 1,500. I mean, Oren will probably be, I don't know if you guys know exactly, but with SMARC-Seq2, you get up to, say, 3,000, 4,000 transcripts per cell. And this is really reproducible, and our best cells show 6,000, more than 6,000, 7,000 transcripts. So with SMARC-Seq2, you can really work. The problem is that each 96-well plate is about a $10,000 cost. This is the sort of the bottleneck with these experiments. So we don't pay for them for those at the moment, but once the broad will start sponsoring this, it will be impossible. So people are now doing microfluidics on thousands of cells with very, very low coverage, bad data, so you really have to combine a lot of things and do really fancy statistics to get your fit. But with SMARC-Seq2, the data is great, but it's really still very expensive. But I hope it's coming down.
Two questions. First, what do we know about the transcription pattern of Huntington gene in other brain cells, not the gabaergic striatum cells? And if there is a difference, do we know anything about the regulation changes?
Right. So again, I started by saying that one of the puzzles is that we don't know why these neurons are affected and others are not. So the simple answer would be because Huntington is expressed in these neurons and not in others. Not only is Huntington expressed in all neurons of the brain, it's expressed in all cells of the body. It's a ubiquitous protein that is basically expressed everywhere in all developmental stages, in all cells. So it's really strange, and this is pretty much the case with all these polyQ. So we have absolutely no clue, and this is why I think it's a fertile ground to ask questions, because there are no answers.
I have one question. I hope it's not too wide, but is there at this point some vision for how some of these diseases might be treatable?
So just as an historical side note, the mutation of HD was identified in 1993, and if you look at the literature of these couple of 2 years, it was sure that you'll have a cure now, because the gene, they thought they knew the pathway, everything was sorted out, and now we'll have the cure. Not only is the cure not available, there is not even a symptomatic treatment, nothing. So there are basically, two options that are kind of futuristic, but I think doable. One is gene therapy, and there are two ways to treat this from a conceptual point of view. One is to target the DNA, but you don't have to. You can actually target the RNA, because if it's a dominant negative mutation, it means that if you just lower the amount of the protein or RNA, you can probably treat the disease or the symptom. So if people will be, and this is something that I'm now starting to entertain this thought, if we use CRISPR to target RNA, and this just came out in Nature like last week, you can actually do it sort of in a safe mode without affecting the DNA. The problem with using gene therapy with DNA-directed viruses is that you're going to mess out, things that you don't want to. But directing RNA is one optional. So I think either by even siRNA or shRNA or CRISPR-Cas-targeted RNA I think this will be one avenue. The other, of course, neurodegeneration or degeneration in general. Of course, all the hype about stem cells is that one day we'll be able to treat, diabetes type 1, liver, heart, kidney, all the degenerating tissues with stem cells. Why not the brain? I can tell you why not, because we don't understand enough to treat the brain. But, 30 years down the road, or maybe I should say 70 when I'm not around, so maybe then, I don't know. I think that there is now, the EU just sponsored a very, very big consortium with 30 Parkinson's patients, which are basically getting injections of pluripotent stem cells inside their brain, inside the substantia nigra. And I don't think it's going to do much. I'm the skeptic, but we'll see. Yeah. Time, it's a waste of.
There is a solution long term that you can eliminate the birth of these kids.
Yes, of course, for these diseases, but for neurodegeneration or degeneration in general, yeah. Yes. Diseases.
Many mothers are going through this.
So there are diseases that have been also eliminated, like Tay-Sachs, for example, because it was a Ashkenazi, disease, and they now do shidduch. They almost eradicated completely this disease. It's amazing that the religious.
All you need is to obey the rabbi and tell you whether
exactly. No,
this girl is good for them.
I think this is an amazing social experiment where Orthodox Jews were able almost to eradicate the disease just by marrying the correct person. But PGD can eliminate disease. The problem is that many still, we see it with the Machado Joseph, we know the patient, and a lot of them are too shy, they don't want to be bothered. And actually, one of the persons we really got involved with, they tried, and they couldn't get IVF to work. And eventually they had two beautiful twins that are just by natural, and luckily they are both healthy. But this was, a painstaking exercise. So it's not as easy as it sounds. But there is hope. On this happy note, I end. Thank you.